Bismillahirrahmanirrahim. I am Dr. Muhammad Rafiq from Cholistan University of Veterinary and Animal Sciences, Bahawalpur, Pakistan. Today we will learn about the enzyme kinetics, the study of chemical reactions rate that are catalyzed by enzyme. As we know, the enzymes are the biological catalyst which enhance the rate of biochemical reactions. It is an expression of chemical reaction in mathematical terms. It could also be defined as the quantification of an enzyme activity in terms of the rate of conversion of substrate to product based on time or enzyme concentration. The classification of chemical reactions. Chemical reactions can be classified into three: zero order reaction, first order reaction, and second order reaction. First of all, we will discuss about the zero order reaction. A chemical reaction wherein the rate is independent upon the concentration of the reactant or the substrate molecule. That is called the zero order reaction. It means such kind of reaction in which the rate is independent upon the concentration of reactant molecule. In general reaction, we can see this is the substrate molecule or the reactant molecule which is converted into the product P. The concentration, the rate of the reaction or the velocity V of the reaction, that is equal to K into substrate concentration naught. V is equal to K. K is the rate constant. It means in zero order reaction, the rate or the initial velocity of the enzymatic reaction, that is independent upon the substrate concentration. The S naught, that is equal to 1, it means V is equal to K. It means in zero order reaction, the velocity or the rate of reaction is constant. Now we will discuss about the first order reactions. A chemical reaction wherein the rate is dependent upon the concentration of one reactant molecule. That is called the first order reaction. In first order reaction, we will see in general reaction, this is the reactant molecule, the product, the reactant that is converted into the product. In first order reaction, the rate that is directly proportional to the substrate concentration of one reactant molecule. The V, that is equal to K into substrate concentration. K is the rate constant. In here, the reaction rate or velocity is the rate of the formation of product P. Also, the reaction rate or velocity is directly proportional to the reactant or the substrate concentration. It means in first order reaction, as well as the substrate concentration increases, the rate of the reaction also increases. In second order reaction, that is defined as a chemical reaction wherein the rate is dependent upon the concentration of two reactant molecules. That is called the second order reaction. In general reaction, we can see the two substrate molecule, one substrate, second substrate. This is the product. They are converted into the product. It means in such kind of reaction, the rate is directly proportional to the substrate concentration of one molecule and the substrate concentration of other molecule. V is equal to K into substrate concentration of one molecule, substrate concentration of other molecule. V is equal to K substrate concentration square because the product of substrate concentration will be added up. The reaction rate is the rate of formation of product. Usually when we ask the rate of reaction, it means the rate of formation of product. Now, in next slide, we will see all enzymes that obey the Michaelis-Menten kinetics graph of initial velocity against the substrate concentration gives the rectangular hyperbola. In this graphical representation, we can see the substrate concentration is along the x-axis. The initial velocity V naught or the rate of enzymatic reaction is along the y-axis. As well as the substrate concentration going to increase, the rate of enzymatic reaction also going to increase. In this hyperbolic curve, we can see up to certain limit, up to this point, as well as if we increase the substrate concentration, the rate also going to increase. It means at this substrate concentration, the rate is this one. At this substrate concentration, the rate is this one. At this substrate concentration, the rate is maximum. After that, if we increase the substrate concentration, the rate did not increase anymore. At this substrate concentration, the rate is same. At this rate is same. It means in the beginning, if we increase the substrate concentration, the rate is directly proportional to the substrate concentration. As well as we increase the substrate concentration, the rate is also increased. It means from here to here, from this point, this is the, from this point to this point, this is the first order kinetics. It means the rate depends upon the substrate concentration molecule. And after that, from this point to this point, the rate is independent upon the substrate concentration. If we increase the substrate concentration, the rate did not change. It means now the substrate concentration become saturated. This is the zero order kinetics. Now we will derive the Michaelis-Menten equation. In an enzyme catalyzed reaction, a free enzyme E binds its substrate S to form an enzyme substrate complex, which either undergo further transformation to yield the final product P and the free enzyme, or break down via a reverse reaction to form the free enzyme E and the substrate S. This is the general reaction. Herein, we can see this general reaction is divided into two portions. This is the first portion in which the enzyme bind to the substrate and they form the enzyme substrate complex in forward reaction. And the rate constant of the forward reaction is K1. In backward reaction, the enzyme substrate complex that form free enzyme and the substrate molecule, and the rate constant is K2. In other words, the second portion of this reaction is this one, which is irreversible, in which the enzyme substrate complex that is converted to product and the free enzyme, and the rate constant is K3, which is irreversible. And this first portion is reversible portion and this is the irreversible portion. We will discuss both the portion and derive the equation. Now, rate of formation of enzyme substrate complex in forward reaction. In this forward step, we can see the rate of formation of enzyme substrate complex means this product of first step that the rate is directly proportional to the concentration of reactant molecule. The reactants are free enzyme and the substrate. Rate of breakdown of enzyme substrate complex, this reverse back step, the rate is K2 that is equal to enzyme substrate concentration. In this case, the reactant is enzyme substrate molecule. This one. This is converted to this one and this is converted to this one. Rate of formation of enzyme substrate complex. This is formation which is K1. Enzyme concentration, substrate concentration, equation 1. And in equation 2, the rate of breakdown of enzyme substrate complex. Enzyme substrate complex breakdown in first step into E plus S, in second step into E plus P. Now we can write K2 enzyme substrate complex plus K3 enzyme substrate complex. This is equation 2. Enzyme substrate complex is the common value. That's why we can write it as a common value. K2 plus K3 into enzyme substrate concentration. This is equation 2. When the reaction is at equilibrium state or at steady state, then the rate of formation of enzyme substrate complex become equal to rate of breakdown of enzyme substrate complex. By putting the value of formation and the breakdown of enzyme substrate complex K1 into enzyme concentration, substrate concentration, that will be equal to K2 plus K3 into enzyme substrate concentration. By rearranging this value, we can see the enzyme substrate concentration become equal to enzyme concentration, substrate concentration divided by K2 plus K3 divided by K1. Now, this is equation 3. K2 plus K3 divided by K1, that is equal to Km. Km is the Michaelis-Menten constant. By putting the value of Km into equation 3, we can get enzyme substrate concentration, that is equal to E into S divided by Km. This is equation 4. Total enzyme ET, that is equal to free enzyme plus the enzyme which bind with the substrate to form the enzyme substrate complex. So we can write total enzyme concentration, that is equal to enzyme concentration plus enzyme substrate concentration. This is equation 5. And the enzyme concentration from here, we can write enzyme concentration, that is equal to ET minus ES. This is equation 6. By substituting the value of enzyme concentration from equation 6 into 4 above we have. By putting the value of enzyme concentration into equation 4, we have enzyme substrate concentration, that is equal to ET, total enzyme concentration, minus ES concentration into substrate concentration divided by Km. This is equation 7. By rearranging the equation 7, we can get enzyme substrate concentration, that is equal to ET into substrate concentration minus ES into substrate concentration divided by Km. This is equation 8. We can also write enzyme substrate concentration is equal to Et into substrate concentration divided by Km minus Es into S concentration divided by Km. This is equation 9. Now, we can shift this negative value into the other side of the equation. Then we can write enzyme substrate concentration plus Es into S divided by Km, that is equal to Et into S divided by Km. This is equation 10. By multiplying Km on both sides of the equation 10, Km into Es, Km into Es into S divided by Km, Et into S divided by Km into Km, Km will cancel out, this Km will cancel out. So Es into Km plus Es into S, that is equal to Et into substrate concentration. This is equation 11. Es is the common value in this equation. That's why we will write this as a common value. Es concentration into Km plus substrate concentration, that is equal to Et into S. This is equation 12. Enzyme substrate concentration, that will be equal to Et into S divided by Km plus substrate concentration. This is equation 13. We can also write enzyme substrate concentration, that is equal to Et multiplied by substrate concentration divided by Km plus substrate concentration. This is equation number 14. From Macquarie's-Menton model of kinetics, we know the initial velocity, that is equal to K3 into enzyme substrate concentration. Hence, enzyme substrate concentration, that is equal to V divided by K3. This is equation 15. Also, we can write Vmax is the maximum velocity. Maximum velocity is equal to K3 into total enzyme concentration. Hence, we can write Et, that is equal to Vmax divided by K3. Now, substitute enzyme substrate concentration from equation 15 and total enzyme concentration from equation 16 into equation 14. This is the equation 14, enzyme substrate concentration, that is equal to total enzyme into substrate concentration Km plus substrate concentration. Enzyme substrate concentration is equal to V divided by K3, that is equal to V max divided by K3 into substrate concentration divided by Km plus substrate concentration. This is equation 17. By multiplying K3 on both sides, we can write V is equal to V max into substrate concentration divided by Km plus substrate concentration. This is equation 18. By rearranging, we can also write V is equal to V max multiplied by substrate concentration divided by Km plus substrate concentration. This is called the Macquarie's-Menton equation. When substrate concentration is much less than Km, when this value is much less than Km, the rate of the reaction, that is proportional to the substrate concentration. It means when this value is very less, then if we increase the substrate concentration, the rate will also increase. And the rate of the reaction is said to be the first order with respect to substrate concentration. First order means the rate depends upon the substrate concentration. When the substrate concentration is much greater than the Km, the velocity or the rate of reaction is constant and equal to V max, maximum velocity, then the reaction rate is independent upon the substrate concentration and is said to be zero order reaction with respect to substrate concentration. It means when the substrate concentration is much greater than Km, then the rate is independent upon the substrate concentration anymore. Then this is called the zero order reaction. This is the end of the video. Thank you very much for watching. Please don't forget to subscribe our CUAS channel. Thank you very much.